Okay, so welcome everyone for the Plant Postdoc seminar series. This is our 2021 fall seminar series, and we are happy to have two exciting talks today. So the format of the seminar is our speakers will talk about their recent work, either a preprint or recently published work for 20 minutes, and there will be time for Q&A for 10 minutes. And after both the speakers have presented their work, we'll have an informal chat session, which will not be recorded, and we can just talk to the speakers and the Plant Postdocs leadership team about the general questions about the talks or about any other things about Plant Postdocs or anything about the early career researchers. So without taking much time, I'll introduce the speakers today. So our first speaker is Dr. Guangchao Sun, who was a postdoc in University of Nebraska-Lincoln Center for Plant Science Innovations in James Schnabel Lab, and is currently moved to Mayo Clinic. I overlapped for a short time with Guangchao, and he's an extremely talented scientist, and I'm sure you will realize that by the work that he has done. And he'll be presenting today about a sugar tail told by the omics of a wild crop relative, the seashore paspalum. Yeah, you can take now, Guangchao.
Okay, thank you, Sunil. Well, first of all, I want to give a big shout out to Plant Postdocs. I'm a long time Plant Postdoc follower. I really appreciate all the valuable information regarding career development and job applications. So it's also very honored to be invited to give this presentation. So first of all, my name is Guangchao Sun. I did my PhD in plant pathology, and I joined James Schnabel Lab in 2017. And during the time I worked as a postdoc in his lab, I learned a lot of bioinformatics by working on transcriptomics, genomics, and quantitative genetics work. So I learned a lot. If you are interested in working with this group, just feel free to reach out to Dr. Schnabel. Today I'm going to talk about this sweet story that we unraveled from a mix of a wild crop relative called a seashore paspalum. The scientific name is Paspalum vaginatum, and we also did comparisons with these crop relatives, maize and sorghum. So the sugar I'm going to be talking about is actually drawn here. I don't know if you can see my pointer here. So it is a disaccharide called Trello. So I'm going to let you know like how did we find out this story. So first of all, what is paspalum? It has a lot of nicknames. It has been widely used as a turf grass for coastal golf courses, and if you are from coastal cities, you probably get a chance to see this kind of grass along the seashore. But by no means would you imagine this is actually a close relative to maize and sorghum. The flower is like that. It's kind of beautiful. But if you self it, if you just bag it, you won't get seeds because it's a heterozygous, self-incompatible. So it's a obligate outcrossing grass. And one thing that really amazed me by this grass really amazed me is it's resilient to a variety of stresses like salt, cold, drought. They are not a problem for this grass, and it was reported that this grass performs as well as other turf grasses when you supply lower amount of nitrogen. This is the aspect I'm going to be exploring in this project. And what makes it even more important is it is a wild relative to maize and sorghum. So there is always a big question that we want to ask is that, so what can we learn from the wild relative for resilient breeding in crops? So during the time I was working in Schnabel Lab, we were collaborating with JGI and Hudson Alpha team for genome sequencing, assembly, and annotation. Once we got the the genome finalized, we performed some comparative genomics analyses by using more than 6,000 conserved single copy orthologs. We constructed this phylogeny, confirmed that Paspalum is indeed the first sequenced wild relative of maize and sorghum. So the phylogeny, phylogenetic relationship between Paspalum and maize and sorghum is even closer than that between Citaria and maize and sorghum. So the genome of this species is really meaningful because we can learn a lot about resilience of maize and sorghum relatives. Those numbers, so it shows that Paspalum diverged from maize and sorghum at 28 million years ago. And those numbers on the branches indicates the gene family is undergoing either expansion or contraction. So we also identified a set of gene families specifically expanding in Paspalum. I'm going to touch a little bit more on that later. So as I mentioned, I was interested in how Paspalum is dealing with nutrient stress. So we performed this experiment where all the 3 species are growing under these 3 nutrient conditions, either full, which means all the nutrients are supplied, or nitrogen starvation or phosphorus starvation condition, and measuring the biomass accumulated in the above ground. And as you can see here, maize and sorghum, visually, you can see it's really obvious that the growth is reduced. But Paspalum did not show any significant reduction, and also is reflected by biomass accumulation. There's no significant change in Paspalum, but there's a big reduction of biomass accumulation in maize and sorghum under nutrient stress condition. But on the top of this different phenotype of the above-ground phenotype, we actually observed a conserved root elongation across the 3 species under nitrogen starvation condition. However, there's only a significant elongation in sorghum under phosphorus starvation condition. So these phenotypes, so I would say this smaller phenotypic plasticity observed in Paspalum is expected if Paspalum is indeed resilient to nutrient stress. But there's a big question here is whether or not Paspalum was indeed stressed by the conditions we conducted in this experiment. Because the impression of Paspalum is that it's a wild relative, it grows generally smaller and slower, so probably does not need that much nutrient. So the experimental conditions we use might not be sufficient to stress the species. Therefore, we see this no change of phenotype. So the most direct evidence that we can address this question is to quantify the nitrogen or phosphorus in the shoot tissue under nitrogen or phosphorus starvation condition. And we did observe a big reduction in nitrogen and phosphorus under nitrogen or phosphorus starvation condition, even though the Paspalum showed the least reduction of phosphorus under phosphorus starvation condition, but the reduction is there. So therefore, but we did see a comparative level of reduction of nitrogen under nitrogen starvation condition. Therefore, we just focused more on the nitrogen starvation condition in the later study. Also, plants tend to accumulate starch in the shoot once they are stressed by nutrient. So we also examined the genes encoding the enzymes that promote starch accumulation or promote starch degradation. So the expression pattern of these genes actually is indicating a tendency of starch accumulation in Paspalum shoot, indicating that Paspalum was indeed stressed and is trying to deal with that nutrient stress. So with all of this evidence showing that Paspalum is indeed stressed by nutrient deficiency in our experimental conditions, so we were safe to proceed for more experiments. So the first thing we did was to profile the primary metabolite to see the metabolic response of the 3 species under the nutrient stress conditions. So here I'm showing you the metabolite fold change under either nitrogen or phosphorus starvation condition comparing to full condition as a reference. So the more red you see here means the metabolites is more increased under nitrogen starvation condition comparing to that in full condition. So the more blue you see means indicating the more decrease of this metabolite under nitrogen starvation condition comparing to full condition. And if you see gray, means this metabolite did not show any significant fluctuation. So the overall impression you can get from this panel is pathplum did not show a big difference, a big change in primary metabolism. And we also see that maize and sorghum are pretty much similar because they are phylogenetically similar as well. And we also observed a conserved response of metabolites, conserved a change of metabolites across the 3 species under nitrogen starvation condition. But one thing I want to point out here is that the allantoin, it is reduced consistently across the 3 species, and allantoin is actually a storage form of nitrogen when plant is storage form of nitrogen in root. So when plant encounter nutrient stress, allantoin can be converted into mobile form of nitrogen to support or to reduce to neutralize the nutrient stress. So that also gives another layer of evidence saying these plant, all the 3 species are stressed and they are trying to deal with the nutrient stress. So the similar situation was observed under phosphorus starvation condition. So pathplum show overall less fluctuation, and pathplum, maize and sorghum are pretty much similar. But you see the overall all the metabolites are reduced. But there's a striking accumulation of Trellos really caught our eye. And this was also observed under a nitrogen starvation condition. And statistically, it is significant in pathplum, not in maize and sorghum. So we know that Trellos has been reported to be involved in stress response in plant species, fungal species, or even mammalian species. So we just decided to pay more attention on that because we did not know much about Trellos, potentially involved in nutrient stress condition, nutrient stress response. So next we performed a transcriptomic analysis with all the samples, and we identified the differentially expressed genes under nitrogen starvation and phosphorus starvation conditions using full condition as a reference. It turned out that most of the differentially expressed genes are syntactically conserved. By syntactically conserved conservation, I mean the genes are conserved in both sequence and the location, which means these genes are more likely inherited from their common ancestor. So there's a common idea that common thoughts you have is that if these genes are syntactically conserved, that means they are not changed after divergence from their common ancestor. Probably their behavior will be also conserved under the same condition, but actually it's not. If you look at those syntactically conserved genes, they did not. Their response to either nitrogen starvation or phosphorus starvation conditions are sufficiently diverged. So in this situation, we decided to focus on 2 cases. One is like this set of genes. So the set of genes that are really conserved in terms of their response to the same nutrient stress condition. And so these sets of genes very likely represent the core genes that are responding to nutrient stress. And another set of genes we are interested in was the genes that make pathplum unique. So the genes that respond to nutrient stress only in pathplum. We examined the processes or functions overrepresented in these 2 sets of genes. And we found that those processes are very likely the core metabolite, the core processes responding to nitrogen starvation or phosphorus starvation condition. So these are the genes. So these set of genes are the conserved ones. And also, so the genes responding to nutrient stress only in pathplum, were also enriched in these processes. We noticed that there are a lot of processes related to like antioxidation or transporter activities or gene expression regulation. So these might be the processes really analyze the resilience of pathogenic to nutrient stress. As I mentioned before, during the comparative genomics analysis, we identified a set of gene families that are undergoing gene copy expansion, specifically in pathogenic clade as a branch. So we found that actually there are a lot of gene family members also responsive to nutrient stress, which means pathogenic might have been experiencing nutrient stress in its natural niche, and these gene families are expanding to help pathogenic to overcome it during the evolution. So now come back to the Trello story. So now the primary metabolite profiling provided one layer of evidence that Trellos might be the reason why pathogenic showed unique resilience to nutrient stress. But how about it at the transcriptomic level? So here I'm showing a metabolic, Trello's metabolic pathway in brief. So it starts from 2 molecules of glucose, and the Trello 6-phosphate synthase will convert 2 molecules of glucose into Trello 6-phosphate, and then T6P can be converted into Trellose by Trello's 6-phosphate phosphatase. And then Trello can be degraded into glucose by Trellase. If you think about, when we think about the Trellose accumulation, we can think of the potential mechanism in 2 ways. One is the upstream production process is very active, right? And on the other hand is that maybe the degradation process is very inactive. There is a big redundancy of genes encoding these two types of enzymes involved in the production of Trellose. So it might take much longer time for me to explain, but there is only one copy encoding Trellase. So we just went ahead and checked the expression of this gene. We found that the expression level of this gene is surprisingly lower in pathogenic than that in maize and sorghum. So as I mentioned, probably the degradation of Trellose might be very slow in pathogenic, which might be the reason why Trellose experienced accumulation. So we also found that actually the genes involved in Trellose metabolic pathway are undergoing faster sequence change during the evolution, as reflected by the distribution of the KKS ratio. So that means maybe Trellose metabolic pathway is doing something, has been playing a role in giving pathogenic the resilience to harness hardships in its environment. So now I provided 3 lines of evidence showing that Trellose metabolic pathway might be a potential mechanism underlying its resilience. Then the question is, can we recapitulate the pathogenic phenotype? As I mentioned that the Trellase expression is pretty high in maize and sorghum, without very effective or simple genome editing tools in hand at that time, we decided to probably apply some chemical genetics approach to see if we could reduce the Trellase activity to reduce the Trellose degradation. So we found that there's a Trellase specific inhibitor called the validomycin A that can just inhibit Trellase activity. So that we try that to see if we could induce internal Trellose accumulation in maize and sorghum, and see if we can recapitulate the pathogenic phenotype. And the answer is yes. We see that when we just grow the plants. Here I'm only focusing on maize for the sake of simple understanding. So when you grow the plants under full and natural starvation condition, we do see a visually noticeable size difference between the groups without validomycin treatment and with validomycin treatment in maize. But there's no significant change in pathogenic. One question we need to answer is whether it really caused the accumulation, internal accumulation of Trellos. So the answer is yes. There's a significant increase of Trellos with rhodamycin treatment, and we see that the site is actually reflecting a more larger above ground biomass accumulation in maize, but not in past plum, as well as shoot to root ratio. So the higher the shoot to root ratio, the more biomass was allocated to the shoot, which is a desired trait for crops. All of that data was shown for the seedlings, the 3-week-old seedlings. So how about the adult plants, right? So we just keep adding rhodamycin A treatment to the plants until it starts flowering, and we examine the biomass. We see that there's still a significant increase of biomass in the plants with rhodamycin A treatment. And after flowering, we see actually those plants at the same time. The plants gives larger and better reproductive organs. So then the question is, what is going on? What is the molecular mechanism underlying this rhodamycin A mediated biomass increase? So based on the previous knowledge about this, the Trello's metabolic pathway involved in the stress response in plants or other organisms, we know that there's a connection between Trello 6-phosphate and SNRK1, which this pathway or this signaling axis is balancing carbon and nitrogen sensing, and also to regulate the cell growth. So, but thinking about the Trello's metabolic pathway, what is the effect of Trello's accumulation, right? So we propose maybe Trello's accumulation will impose a negative feedback effect to Trello 6-phosphate, and which actually directly regulates SNRK1 activity. So we examined some key enzyme expression, the expression level of the genes encoding these key enzymes in maize. So the expression pattern change is showing us that Trello 6-phosphate might be reduced, and also the SNRK1 subunit is upregulated, which means SNRK1 activity might be actually increased, which will in turn promote autophagy. And we also observed all of the nitrogen transporters in root are upregulated with rhodamycin A treatment, as you can see here. So that is indicating, okay, in root, the autophagy is actually upregulated with the rhodamycin A treatment, and also as well as the transporters. That will definitely help the plant to first be more efficient in the nitrogen uptake, and also more efficient in nutrient recycling and reallocating. Is that true? And the answer is still yes. Like we examined the conjugated ATG8, which is a marker of autophagy activity in both 3-week-old root and the 1-week-old root, and you see that there's more ATG8 PE, which this band, the lower band is shown here. And also we observed the more of the phytosome stained by MDC in the root cells in the 1-week-old seedling. So that gives us the evidence showing maybe rhodamycin A treatment is actually causing the autophagy activation. So we performed a non-experiment to confirm that if we inhibit autophagy by 3-MA, the rhodamycin A is no longer able to cause the increase of biomass. Therefore, we could further confirm that rhodamycin A treatment mediated biomass increase is indeed autophagy dependent. So at the last, I give you a summary of this project. We have this all of this data is showing that first of all, past plum is really resilient to nutrient stress, and the Trello's metabolic pathway might be playing a very important role in rendering this kind of resilience. And we can simply apply this chemical genetics approach to try to transfer this resilience to the crops. I mean, there's still a lot of questions to be answered, a lot of work to be done in the future. But at least we somehow, gives you these insights that we probably can do something, can make this easier for crop resilient breeding. So at last, I want to give my big thanks to Schnabel Lab, my postdoc supervisor James Schnabel, and all the lab members that helped with this project. And Toshihiro Obata Lab helped us with the primary metabolite profiling, and JGI Thousand Alpha team helped with the genome sequencing assembling and annotation. So UNMC Sequencing Center helped us for RNA-seq, and also a lot of analyses were done in the Holland Computer Center at UNL. And also big thanks to all of these funding reagents. So with that, I'm happy to take any questions you have.
Thank you, Guangchao. It's a very nice story. If there are any questions, please unmute yourself and you can ask the question to Guangchao. Okay, maybe I will start with the first question. So Guangchao, I think you mentioned about this, but I might have missed this. So you said you are trying to see if the nitrogen or the phosphorus starvation that you are giving is a stress to the paspalum. So I was wondering if that is related to how maize and sorghum behave in the field. Do you know what concentration of nitrogen or phosphorus is stress for maize growing in the field? And did you compare that to the stress levels that you are giving?
Yeah, that's a nice, good question. For the experiment, we had this concern at the very beginning when we performed the experiments, and in this experiments conducted in greenhouse, we were actually just completely depleted the nitrogen from the nutrient solution. So when I say this is a nutrient solution without nitrogen, there's no nitrogen in the solution. So basically, when we plant the seeds in the soil, actually the soil is only the media we used actually only contains a small amount of new soil just to support the early growth of the seedlings. So if you just grow the plants for a longer time, there will be a time point, all of the nutrients, all of the nitrogen or phosphorus will be used up. So the plant will be undergoing nutrient stress, but we just need to quantify the nitrogen and phosphorus to make sure that paspalum was indeed stressed. Now go back to the field question. We in the field, usually you still give nitrogen, you still supply nitrogen, but the lower, if you want to stress the plants, you give a lower amount of nitrogen. Maize and sorghum are sorghum will be better. I mean, maize is pretty sensitive to nitrogen starvation. I cannot recollect the exact concentration you can stress maize, but if you just reduce half of the normal amount of nitrogen you supply to maize will be stressed. It also depends on which stage did you apply the nitrogen starvation. But the flowering phenotype was really can be really affected if you wait until the flowering season. So maize is pretty sensitive. Sorghum is a little bit better, but still, if you just reduce the amount of nitrogen supplied, sorghum can be stressed as well.
Okay, thank you. There is another question here in the chat by Takehiro. Great talk, Guangchao. Did you test chemical inhibitors of TOR in paspalum? Example, combined treatment of Val A and AZD-8055.
That's a good question. We actually, in the first batch of experiments, I used the rapamycin, which is a specific inhibitor of TOR. but there are some studies in Arabidopsis showing that the plants are way less sensitive to rapamycin or TOR inhibitors than mammalian or fungal cells. So yeah, I did not do that, but I see what you mean. If we inhibit TOR, first of all, we can inhibit growth, and probably we can also activate autophagy. But we did not know like how pathogen will respond to TOR inhibitors. But right. I mean, it's worth trying. It's worth seeing. Don't know if it's sensitive to this inhibitor that you mentioned, but it's worth trying.
Just to continue, there is a TOR independent and dependent autophagy. ER stress is TOR independent autophagy, and nutrient starvation is TOR dependent autophagy. So that's why I had certain noise in the definition of autophagy dependent, because it can be TOR independent or dependent. At least in
exactly. So based on the previous studies, we infer that it's very likely this autophagy is TOR independent. First of all, because when we add validomycin A, so if you look at this last slide right now I'm showing on my screen, validomycin A treatment caused a larger size of plant, which means the plant is growing larger or growing faster. So this cannot be achieved when TOR is inactivated. When TOR is inactivated. But validomycin A treatment is indeed causing autophagy in root, and we see some studies conducted in the human, I think it's a mammalian system, showing that Trello also cause autophagy in a TOR independent manner. So it probably is is likely the same case in plant, but I think this what you propose is something that we should do, or I mean we are interested in doing. It depends on whether or not these plants are really sensitive to the inhibitors. Yeah.
Yes, the second generation como ACD or TOR one is better to use than rapamycin. Rapamycin is not a good idea using.
Yeah. That's a really good idea. Thank you.
Yeah, thank you for the questions. If we have more questions, we have time after the second talk. We can talk to both the speakers in the informal session. So again, thank you to Guangchao for a wonderful talk. Now we'll move on to our next speaker. Our next speaker is Dr. Ankita Pandey. She is a postdoc in Pohang University of Science and Technology in South Korea. And today she'll be talking about bacterial wilt resistance in wild relatives of tomato.
Yep. Can you hear me at least?
Yes.
Yeah. Okay, good. So thank you so much for this opportunity. The work that I am presenting here today is basically my PhD work that I have continued for my postdoc as well. So I will be talking about the identification and functional characterization of Ralstonia solid serum avirulence effector proteins recognized in wild relative of tomato. This work is basically one of the works that we have basically done in our lab, wherein we concentrated on the effector biology. It's only a part of my PhD work where I am only showing the tomato part, but we have similar study also in pepper, which will be published soon. So basically, I'll start with just a bit of introduction to the plant immune system. So as you know that in plants, in nature, plants are attacked by different sets of pathogens, but be it a bacteria or a nematode, fungus, and so on and so forth.
Sorry, do you have your slides?
Oh, can you not see it? I thought
No,
I
just sent you a message. Yeah.
Oh, really? Okay, let me just try. Can you see it now?
Yeah, we're seeing your screen now. Okay, now the slide came up.
Yeah, okay. Sorry, I thought
Yeah.
it was already visible.
Good, thanks.
Okay, so yeah, so as I was saying, there are different pathogens that attack plants in nature, and these different pathogens have something called as PAMs or MAMs, which are basically molecular patterns which the bacteria or the fungus they introduce into the plant system. And these different PAMs are basically tackled by receptor proteins in plants, and they are called as PRRs. And this activates, their recognition by PRR activates the first layer of the plant immunity, which is PTI or PAM triggered immunity. But on the other hand, all these pathogens, they are also smart. They know how to, get away with PTI. So they have a different mechanism wherein they have developed a certain a structure by which they can introduce the effector molecules into the plant cell. And these effector molecules basically can, if they go unrecognized, they can cause disease in the plant. But to tackle the effectors, plants have got R genes or NLRs as we call them, and they can directly or indirectly recognize these effectors, and that triggers the effector triggered immunity or ETI. So my today's presentation will be mostly concentrated on ETI. So before I start with my presentation, like the work that we have done, I just want to give you a brief introduction to what is resistance and what is susceptibility and how it's decided. So when it's basically the gene for gene model that was proposed by H and Floor, wherein when the pathogen that attacks the plant has a virulence effector, so whether or not, whether if the plant has a resistance gene, then the plant becomes resistant because the resistance gene in the plant recognizes the effector, virulence effector from the pathogen. On the other hand, if the plant lacks the resistance gene, doesn't matter if your pathogen has a virulent effector or not, because it will not be recognized here and the plant will be susceptible. On the other hand, if there is no virulence effector in the pathogen, doesn't matter whether your plant has a resistance gene or not, it will still be susceptible. So there are a lot of virulence genes that have been recognized, that have been identified previously, and their corresponding disease resistance genes have also been identified. But today we are talking about the pathogen, one of the very economically important pathogens that is not very common to study. It's called Ralstonia solanicerum. It causes bacterial wilting in plants. So the mechanism of this wilting is that it has it's like it stays in the soil for unfavorable conditions, and when its right humidity and warm temperature is met, then it enters through the root system of the plant and produces exopolysaccharides, which will block the nutrient flow in the xylem, and that blocking of the nutrients causes the bacterial wilting, and this kind of wilting is irreversible and ultimately fatal for the plant survival. The biggest species of plants that are affected by Ralstonia are those of the solanaceous species. So it's a huge host range, somewhere around 200 plants. But the most important crops that are affected are that of tomato and pepper, including potato as well. Why is Ralstonia so interesting to study? So after Pseudomonas, Ralstonia is considered as the second most economically important pathogen. it is very heterogeneous in nature. So they have the classification of Ralstonia is such that it has been divided into various phylotypes based on the origin of the, based on its origin and the host plant of that is being attacked by Ralstonia. So the type 3 secretion system, or the effectors that we are talking about here, the effector repertoire of Ralstonia is extremely large. In normal cases, for example, Pseudomonas or Xanthomonas, we generally observe that those kind of pathogens have 30 to 40 effectors in their repertoire. But Ralstonia, it has over 100 type 3 secretion effectors, and they are generally called as injected proteins, or Ralston or RIPs as we call them. And there are generally 60 to 70 effectors in each Ralstonia strain. Another very important aspect of Ralstonia is generally in US or Europe, Ralstonia is considered as a bioweapon. So it's a select agent, so you wouldn't see a lot of labs working on it. And so even though it's a very economically important pathogen, but there isn't much information available about the virulence effectors that are recognized in various Solanaceous crops. So that was one reason to take up this study. So in this particular work, we have generally concentrated on Korean strains that are found from different parts of Korea. So we had a collection of over 30 Korean isolates of Ralstonia, which were identified and collected from different hosts like tomato or pepper or potato from various different parts of Korea. And this work so we collected these strains, we sequenced them, we have all the effector repertoires of all these 30 strains, and that work was already published in last year from our lab. So what we did was to start with, the first step of the work is to do a screening of the different tomato accessions that we wanted to study. Why tomato? Because tomato is the second most valuable vegetable crop. Bacterial wilt in itself can cause somewhere around 90% of crop loss in tomato. General practice, how tomato is grown these days, is basically you have a resistant stock and then you put your commercial whatever cultivar graft on the resistant stock, and then you generate the crop. But that is quite costly, and if you have a resistant elite cultivar, then life becomes easy for a farmer. So that was the whole idea behind using tomato. So what we have done here is basically we had two accessions of tomato. One is LA4504, which is Lycopersicum, Solanum lycopersicum. And the other one is LA2093, which is Solanum pimpinellifolium. Lycopersicum is basically a commercial cultivar here, and pimpinellifolium is a wild relative of tomato. Using pimpinellifolium, there are a lot of studies where they have found different, traits like genes for different traits for abiotic stress to biotic stress and so on and so forth. As far as plant immunity is concerned, one of the major genes called as PTO, which is against bacterial speck, which is Pseudomonas syringae, was identified from Solanum pimpinellifolium. So pimpinellifolium is the most closest relative of modern tomato, and that was the whole idea behind using pimpinellifolium for this study. And so we had these two accessions, and we had all the 30 strains of Ralstonia, and we basically grow these plants for a month old, and then we infect them with the 30 Korean isolates one by one to find and observe symptoms over like 10 days or so. And then we can find some very striking phenotype. For example, here, as you see, this Lycopersicum accession is very, very susceptible against this PE9 or 18 or 57, these strains. Whereas on the other hand, the Pimpinellifolium accession is quite resistant against all these strains. So it's quite a differential phenotype that we observed. And this was observed over a period of repetitive screening. Then on the other hand, we also found some strains like PE1, PE39, or TO63, which were both virulent in Lycopersicum or Pimpinellifolium. So there we had two different kinds of phenotypes. One was a kind of strain that shows differential phenotype, and then there were strains that showed completely similar virulent phenotype in both accessions. So then from here on, we chose a very, very clear phenotype showing strains for a further study, which is PE9 and PE1. So as you can see, when we infect PE9, when we infect the two accessions against PE9, Pimpinellifolium remains resistant while Lycopersicum dies, and then PE1, both of them die. And we quantified it using survival assays over a period of 10 to 12 days to show that how the strains are performing. Then the next step from here on is to find the list of the candidate effectors. So how we do it is that we have, as I told you, we have sequenced all the 30 strains, and we know all the effector repertoires of these strains. So we compare the effector repertoire of PE9 versus that of PE1, and then we found effectors that are clearly present in PE9 but absent in PE1. So then we found these four effectors, which are only present in PE9 but absent in PE1. So these became our primary candidates. And then there were secondary candidates which were present in both PE9 and PE1, but they had less than 95% of similarity. So that's how we came up with the list of effectors that we wanted to test. And so the whole idea behind testing this is to find a virulent effector that's recognized in Pimpinellifolium. So what we did was to introduce the 10 different, all these different candidate effectors one by one into the virulent strain PE1. So as I told you, PE1 strain is the virulent strain here. And so these different strains are basically PE1 containing RIP-A1, PE1 containing RIP-AH, and so on and so forth. And then, as I told earlier, we did an infection assay to confirm the phenotypes. And as we observed, all of these effectors, they were showing virulent phenotype in Lycopersicum. And in Pimpinellifolium, except RIP-J, all the others were showing the virulent phenotype. So basically, RIP-J was showing kind of an intermediate phenotype, not completely virulent. So well, by the way, I want to mention here that for the introduction of these effectors, we used constituted of HARP-B promoter, which is very common for Ralstonia. So all these different effectors are expressed under HARP-B. So then when we observed this intermediate sort of phenotype, we tried to change the promoter from HARP-B to native promoter for RIP-J, and then we tested the phenotype again. And we saw a very, very clear phenotype here. So as you can see, PE1, here is PE1 containing empty vector, which is completely virulent in both accessions. But then there is PE1 containing RIP-J from PE9. And as you can see, the intermediate phenotype that we observed earlier is completely changed into a very strikingly, strong resistant phenotype in pimpinellifolium. So from here, we were able to say that RibJ is basically inducing the bacterial wilt resistance in pimpinellifolium. And of course, we quantified it using the survival assays for over a period of 10 days. So then the next thing that we wanted to test is, what about the natural variation of RibJ? So as I told you, we had 30 different Korean isolates that we were using for this study. So we looked for the RibJ variants from all these 30 strains, and then we came up with these different variations. So for example, compared to PE9 virulent, avirulent strain, we had GMI 1000, which is one of the very representative strains of Ralstonia. And then these were all the variations that we observed in the Korean strains of RibJ, alleles of RibJ. So what we did was basically to analyze whether these RibJ variations are also able to trigger the avirulence or not. We wanted to test that. So basically what we did is we had a PE1 virulent strain, and then we introduced all these alleles of RibJ from different strains into the PE1 virulent strain, and then we basically infected the plants with these transformed strains. And as you can see, P1 empty vector, like earlier, as I showed, they are able to cause disease in both accessions. P9, as I already mentioned, P9 is able to trigger the resistance in pimpinellifolium. But along with P9, we observed that GMI 1000 or PE3 strains, alleles of RibJ are also able to trigger the resistance in LA2093. And then we also observed that there are different alleles of RibJ, that is, for example, P57 or TO28. These alleles have got additional polymorphisms which contribute to the evasion of recognition in pimpinellifolium, and hence we see a strong susceptible phenotype in pimpinellifolium. So if we go back to the previous slide, if you see here, PE3 and GMI 1000 have very little non-synonymous polymorphism here compared to PE9 strain. But on the other hand, if you look at PE1 or P57 or even TO28 alleles of RibJ, you will see that there are a lot of non-synonymous mutations, and then there is additionally a C-term truncation for TO28. So our hypothesis here is that probably one of these polymorphisms or the truncations are contributing to the evasion of recognition in case of pimpinellifolium. So then we wanted to see what is the function of RibJ. So there we found through literature survey that RibJ, along with the POPI2 or other different Ralstonia effectors, belongs to a class of acetyltransferases. It's putative acetyltransferase, basically. So our the literature survey says that all these acetyltransferases generally have a catalytic residue, catalytic triad, wherein there is a histidine, a cytosine, and glutamic acid. So we just compared the known acetyltransferases like Hobsy, or basically from Pseudomonas or Xanthomonas that are already known and studied, along with RibJ, and we found that the two of the catalytic residues, that is histidine and aspartic acid, they're all conserved in both in RibJ, but then the cysteine was replaced by a serine in case of RibJ, while it is conserved in others. So then our next question was, whether these catalytic residues are required for the aberrance function of RIPJ. So to test them, we generated mutants of the catalytic mutants, basically, of RIPJ, wherein we have a H2A mutant and D2A mutant, and we introduced again into P1 strain, and then tested with the infection assays. So as we can see here, P1 empty vector, our control works totally fine, but the H2A or D2A catalytic mutants were, to our surprise, they do not really contribute to the aberrance function. The previous studies that we have seen in case of Pseudomonas or Xanthomonas, we found that the catalytic residues are very much responsible for the aberrance function of those effectors. But in case of Ralstonia, we see that catalytic residues are not really playing much of a role for the aberrance function of RIPJ. And of course, we have done the survival assays. And so then we have another small bit of experiment here, wherein HOPZ5, which is one of the acetyltransferases is known to be autoacetylated on lysine. So we also wanted to see whether RIPJ is autoacetylated as well. And so we had the avirulent and virulent alleles of RIPJ, and we have done just a normal Western blot, and we found that only HOPZ5, as we know that HOPZ5 is autoacetylated, but RIPJ, whether it's a virulent or aberrant allele, they're not acetylated on lysine, which also means that there could be other factors that are, it could be a serine or other residues that are autoacetylated. So that is all we know about RIPJ. So in basic, in summary, we found here that RIPJ is one of the aberrant effectors that are recognized in pimpinellifolium, and we found there are a lot of naturally occurring loss of function mutants of RIPJ, which are able to evade the recognition in the pimpinellifolium accession. And unlike the other counterparts of acetyltransferases, in Ralstonia, the catalytic residues do not really play much of a role in determining the aberrance function of RIPJ. And how is this going to be important for farmers is basically that based on RIPJ, we can identify the corresponding resistance genes. So one of the good things about using these two accessions, Lycopersicum and pimpinellifolium, is that using these two accessions, the TGRC has developed recombinant inbred line. So we have a population of around 148 recombinant inbred lines, which we are currently testing against RIPJ, and based on that scores, like if we get 1-to-1 score, it's more like it could be a single gene or so yeah, it's much of a speculation right now, but we are testing them, and based on our results, we will be able to identify the corresponding R gene in near future. And similarly, if we have identified resistance genes from other crops like peppers, for example, or potatoes, then we can definitely stack them together and then make or engineer bacterial wilt resistant crops which can evade recognition, which can evade the pathogen attack from Ralstonia. So that's about it. Thank you so much for listening to this presentation. If you have any questions, I'm happy to answer.
Thank you, Ankita. Really nice presentation. Again, if you have any questions, please unmute yourself and you can ask your question.
I'm wondering, are you interested in doing more protein biology inference? I mean, even though you don't have to resolve the protein structure, but we have AlphaFold, a new protein structure inference tool. Since you only have 6 variants, you probably can just see if AlphaFold could give you some consistent change in those ones that are virulent. From that aspect, perspective, probably can get some insights on why these variants caused the change of variants or
Yeah, you're right. Actually, the juniors in our lab, a few of them are actually doing that work. So we are trying to have the crystal structure ready for Ripche and several other effectors simultaneously, and hopefully we'll kind of focus on that as well.
Okay, if there are any other questions, you can ask now, or we can move on to the informal session and we can chat with the speakers there. Okay, I guess we can move on to the informal session, but before that, I want to thank both our speakers, Dr. Guangchao Sun and Dr. Ankita Pandey, for their wonderful presentation. If you're a postdoc and if you're interested in presenting in these seminar series, please reach out to us. We have our next session in October and November, and after that we'll be having another seminar series in spring 2022. So now the informal session will not be recorded, so I'll stop the recording and we can discuss with the speakers.